Great. So I'd like to now welcome another one of our partners, Sam Hamedi, who is here. He's the co-founder and the COO of Lifefoundry at this point. And he's going to talk to us today about what they are doing there and how they can interact with the DAMP Lab as well as all of you.
Thank you. Thanks for the introduction, and Doug and everyone in the DAMP Lab for organizing that. Okay, so at Lifefoundry, we built a fully automated robotic platform to help our customers automate and basically help them with their R&D needs. So the architecture of this platform is that we have a central robotic cart, then an arm that's mounted on top of it, and moves around the lab, moves the samples between the different stations. We built a lot of devices ourselves too to make it more easier to have the fully automated lights out foundry. We designed this purposefully to be highly reconfigurable. So if we want to run a different workflow, we don't need to change the hardware as much, just mostly just running a different software and run that different workflow. It's very expandable too. So if we see one equipment that's the limiting factor, we add that equipment, we put it on a bench, and then teach that location to the robot, and we can use it right away. So what difference does it make, right? If it's fully automated or if it's semi-automated? Well, the semi-automation automated foundries, you need to do the experiments one by one. I have the scientists that actually operate the robots and they control the workflows. And it's a lot less streamlined and really relies on the human interaction and their expertise. The way we have built this foundry is that the robots automate the whole process, and the algorithms drive the experiments and the workflows. So this helps with some, there are some obvious first level advantages. There is operational efficiency. So you need fewer people. It reduces the labor cost. Versus a work cell type architecture, you're not locking your equipment in a work cell that may use that equipment for maybe 5% of the time. All the equipment are accessible all the time. You also don't need in a compared to a conveyor belt type architecture. You don't need a separate robotic arm next to each equipment that you have to be able to move the sample from the conveyor belt to the equipment. So this really drives down the dollar per data point, and we think this low-cost, high-quality data is going to really unlock a lot of potential when it comes to the AI models. I think we do have the models a lot. Most people will have the models, but the data is really what's going to unlock a lot of value. It's also very scalable. We can run 24/7, and as I said, when we find a new equipment, a piece of equipment that's limiting the throughput, we can just purchase that and add it and train the position and then use it immediately. So self-driving lab is not something I came up with. I read this paper actually a few weeks ago by the Agile Foundry and Berkeley lab, and what they envision is this basically automated lab tied to an AI algorithm. So the automated lab generates data. The data is given to AI algorithm, and it can make recommendations on the next experiments to try. Then basically you can iterate and close the DBTL cycle, and you can improve very quickly. And then this self-driving lab can connect to all the other self-driving labs or Taco Bell branches, and basically interact with each other all over the world, all over the country, and learn from each other. The data can be shared easily with all the new formats that we are all working to standardize. I can learn from the literature. It can work with traditional labs even to get additional data. Also, we will have some digital twins, so some models of how the biological systems work, and basically this whole system can work together and add a lot of value for the research community. So there are some things that really is hard to do manually or in a semi-automated lab. Examples of that can be, if you want to systematically optimize the titer rate and yield. So how we do this optimization now is that we have a heuristics-based combinatorial method that really relies on in-depth experience of scientists and that specific strain. But if we build this um, 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 and build this model of this algorithm, of this system, and then we have the robots, the AI, they can work together. It can find all the competing and contributing pathways for that particular metabolite, and then it will find all the genes that we could potentially express that can help us with the production of this compound, and then it will identify all these knobs that it can turn, and then you can start doing the optimization with this DBTL cycle. Or if you want to elucidate the genotype-phenotype link, I think it's very difficult to, the search space is very large and it's very hard to find all these, build all these relationships. And what we can do is, we can use this, the only way to actually search it is using this low-cost, high-quality, systematic AI-driven approach. And that's what we're building. We published a paper on this topic in 2019. It was very, it was the first step. It was very simple, optimization algorithm to optimize the lycopene pathway. It was just a simple method, basically, but the search space was explored by the robot. It suggested a few points around the whole landscape, and then we tested a few of those points, and then the algorithm started converging to some hotspots that they're more informative and interesting. So this is what we've been doing. We've been going up and up in the autonomy scale, but that's really the holy grail and what we're building towards. So we have our roots from the University of Illinois. We've been using university labs for a long time. I think this is one of the most underrated infrastructure in the innovation economy. Startups really benefit from these services. We use the Metabolomics Center, the Proteomics Center, DNA services, FACS, flow cytometer, all these equipment. I think that they would have been very expensive and often, prohibitive for us to purchase as a small startup. We had access to IBRL with, large bioreactors that we could use, downstream processing facilities, extensive, analytical lab. Recently, we opened our lab in Bay Area, and we have access to Stanford University, a lot of the similar services and labs that we have access to. But I think what we are very excited about DAMP Lab and what BU has to offer is that it's just more than just one equipment, right? So you can run whole workflows on the DAMP Lab, and I think that's very exciting and can unlock a lot of value for us. So the things that startups can benefit from working with university labs, of course, is CapEx for early stages. VC funding is very hard to come by, and you really need to save it, especially in the earlier stages, because you get a lot more dilution if you have to buy, spend $500,000 buying a piece of equipment. Really have to try to avoid that as much as possible. You can also use the equipment that you don't consistently need by working with these university labs. So you can basically convert your CapEx to OpEx and try to optimize your processes and save some money. And if you see that you needed this equipment, you really need that equipment, you can try to basically use it many times at the university and see what features you like, what brand you like, and then you can buy that brand. So that's also something we really benefited from. The staff at the university are very helpful. We have Courtney here. I'm sure everyone's interacted with her. And then, I think for us, like bioreactor scale up was very difficult. I didn't have that much experience during grad school, and the staff at the IBRL University was very helpful. And also something that we've recently found to be very helpful is using university labs as third-party validations. So some of the customers or even investors, they want us to, validate that we've actually achieved this milestone. And everyone trusts universities, they're third parties, they don't have a horse in the game, and we can have our samples or have our products validated in these labs, and then they really satisfy their, their needs. Also, I mean, we had sometimes issues publishing papers as a as a company, because there is obviously a conflict of interest, and if these results are validated by university lab, I think that also really helps. But we are very excited about what we are excited about working with DAMP Lab is that, as I said, it's beyond a certain, it's beyond just a single equipment. So we can run a complicated workflow. We can, and I really encourage all the startups. I don't know how many startups are here. You can, it's ideal for doing a proof of concept experiment. So instead of going directly to a VC to raise funding, you can do your initial proof of concept here. Even if working with companies like us or other foundries, I mean, it's much easier to de-risk initially using a lab at the university. You reduce the risk and you get a lot less dilution when we go talk to a VC, or when you work with a company like us to basically start doing more of the hardcore R&D. The third-party validation for the other labs that we worked with, they could only validate our final product, but labs like DAMP Lab, they can validate processes too for us. So some of the deliverables for us are processes, and those labs, they really can't validate that and can't help us with that. So we can run our processes on the DAMP Lab platform and just share the run number with the customers, and that can be helpful. Same goes for investors, and when there's an M&A transaction, ideally you don't want to share your core results and data with a potential acquirer, or who, but that probably is the competitor right now. And the using company labs like AmpLab can really help with that. Something else I wanted to mention that maybe a little out of scope for this talk is the problem with reproducibility in academia. I think that a lot of times, of course, the data is not reproducible, and that causes a lot of problem. Often, a small detail is missing, and I think the solution that we've come up with as a community is to ask for the authors to just give us more data. Give us every data that point that you have used in all the figures. But if the data is not reproducible, having more of it really doesn't help as much. But if we have if the journals ask for the DAMP Lab run code and you're publishing your paper, give us all the data, the DAMP Lab run code, give us the add gene acquisition numbers, and in theory, anyone can get the genes from the add gene or the plasmids from the add gene, run the DAMP Lab code, and then you can be able to reproduce the data. So I think that can also be a very interesting standard to have, and really love to see more of that in the industry. So I'll leave it at that. I think at my time, and if anyone has any questions, please happy to answer.
Yeah, you mentioned working with the very integrated part. I want to jump close. Why?
I don't know. I think it's just it's infrastructure. Infrastructure is not that sexy. No one brags about roads and bridges as much. You see this shiny object, and everyone's like, AI is very, everyone's gets very excited, but no one gets excited when someone fixes the bridge in their neighborhood. I think it's just they're just not as interesting, I think, because there's not that much novelty. But this is very important, and I know a lot of other startups that use this very frequently, and I think it's very valuable. I think, and I'm very glad there are more and more universities, adding to this infrastructure.
I'll just say something about that too. I think, let's say you took all the same equipment that DAMP Lab had, but it wasn't a core facility, just sat in the fourth. How would he actually pay for that? How would he build? There's the whole infrastructure, how you interact with the university. Like, is it a grant that a professor has and the PI manage it, and it's run by a bunch of grad students and they're part-time. So I think the key is to get more of these facilities actually full-fledged like service centers and have that infrastructure in place would be a key.
Yeah.
And again, since I'm doing this all the time, while I'm still speaking, I think about it years ago, again, in the same family. At IWBMA, we worked with ACS SynBio to have a special issue of the journal for that workshop. part of that effort, we worked with the SBOL community. I remember at that point, there was also the ability that journal would accept SBOL files, and you could, so it was like you get the journal, and you also upload data. I like this idea that when you run the software that we're creating, you could get a job number, which is kind of this run number. And I think another initiative this community could do is to work with these journals or whatnot and say, these are recognized job numbers. We can leverage the computer science community, how you create these unique numbers and how you archive them and make them available. I think that's a very tangible, doable community objective, just to get run numbers put in.
Yeah, absolutely. I think that would be very helpful. Yeah.